You all kindly keep your microphones muted throughout the session to avoid any disturbances. Good morning, everyone. Respected Principal, Professor Dr. Murnal Bhardwaj, Vice Principal Dr. Arif Anjum, IQAC Coordinator Dr. Shakib Ahmad, Academic Supervisor Professor Imran, Science Faculty in Charge Professor Shahbaz Hussain, and our today's esteemed guest Professor Dr. Hamad Ahmad Shadab and Professor J.D. Sheikh, respected faculty members and my dear participants. A very warm and pleasant good morning to one and all present here. On behalf of Vidya Mandir Commerce and Science College, Malegaon City Autonomous, I say Head of Zoology Department, welcome you all to one day national webinar on innovations and applications in animal biotechnology. Animal biotechnology is repeatedly advanced field that bridges classical biology with sorry for disturbance. Am I audible?
Yes, ma'am.
Animal biotechnology is a rapidly advancing field that bridges classical biology with modern technology, contributing significantly to healthcare, agriculture, conservation, and sustainable development. Through today's webinar, we aim to gain insights into recent innovations, emerging applications, and prospects in today's webinar. Now, I invite Academic Supervisor Professor Imran Ahmad to deliver the welcome note. Please, Professor Imran, sir.
Yes, thank you very much, ma'am. Yes, am I audible?
Yes, sir.
Yes, sir.
Okay. Good morning to all respected guests, distinguished speakers, colleagues, and my dear students. Although our principal, Dr. Professor Mrunal Bhardwaj ma'am, wishes to welcome you all, unfortunately, due to her busy schedule, she is unable to present today. As the academic in charge of the college, I'm very happy to be with you today for this national seminar on application and innovation in animal biotechnology. This topic is not only scientific but also closely connected to our daily lives. Arts, Commerce, and Science College, Malegaon City Autonomous, we believe that education should always serve society. Animal biotechnology shows this connection clearly by improving animal health, ensuring safe food, controlling diseases, and supporting farmers and rural communities. This field also reminds us that scientific progress must be guided by ethics, compassion, and concern for the environment. While new technologies bring many opportunities, they also place a responsibility on us to use knowledge wisely. This seminar provides a valuable platform for experienced experts and young learners to share ideas and learn from one another. I am confident that the discussion will inspire innovation, collaboration, and research that benefits society. I sincerely congratulate to organizing team for choosing such an important and relevant theme and wishes the seminar great success. Thank you very much. Sayyeda madam.
Yes, sir. Thank you, sir. Now, I invite Professor Aksa ma'am to enlighten us about the aims and objectives of today's national webinar. Professor Aksa. Aksa ma'am. Aksa ma'am, are you here?
Ma'am, please go for introduction.
Okay, sir. Now comes the most important part of webinar, that is introduction of our esteemed resource person. I request Professor Parvez sir to kindly introduce our first resource person. Parvez sir. Students are
कैमरा बंद
requested
करा
to
दे
mute their
Respected guests,
microphone.
मेरी तेरा है देख दिख रहा है देख खराब क्वालिटी में आ रहा है
Students
बंद
are
हो गया
requested to mute your microphones. Parvez sir.
yes, ma'am. Respected guests, faculty members, and dear students. On behalf of Mahatma Gandhi Vidya Mandir, Arts, Commerce, and Science College, Malegaon City Autonomous, I, Professor Parvez Akhtar from Department of Zoology, welcome you all to today's webinar. It's my great honor and privilege to introduce today's distinguished guest, speaker, Dr. G. Hamad Ahmad Shadab, Professor and in charge, Section of Genetics, Department of Zoology, Aligarh Muslim University. Dr. Hamad is a distinguished geneticist and geneticist with over 21 years of teaching experience and 27 years of research experience. He earned his PhD in human cytogenetics from AMU, Aligarh Muslim University, and had made significant contribution areas of genotoxicity, nanotoxicity, oxidative stress, cancer biology, and molecular genetics. He has published more than 50 research papers in reputed national and international journals and over 80 research papers at various national and international conferences. Sir has successfully guided six PhD scholars with scholars currently pursuing doctorate, three scholars currently pursuing doctorate research, and has supervised several MPhil and MSc research projects. He has received prestigious honor, including Har Gobind Khorana Best Scientist Award, Environmentalist of the Year award in 2009, and Best Poster Award at International Congress in Canada. He has secured major research grant from DST, SERB, and UGC, amounting to over 1.5 crore, and is fellow of severe reputed scientific societies. He also serves on editorial board of international journal and has held key academic and administrative position at AMU. With his outstanding academic and research achievement, we truly privileged to have Dr. G. Hamad Ahmad Shadab with us today. Please join me in giving him a warm and heartily welcome. Thank you so much. Over to Professor Sayyida ma'am.
Thank you, Parvez sir. Now comes the most important part of webinar, that is introduction of our esteemed resource person. I request Professor Parvez sir to kindly introduce our esteemed resource person.
Hello, Assalamu alaikum, and very good morning to all of you. Am I audible?
Yes,
Yes,
sir.
sir. You can.
So, it's my pleasure and privilege to have some conversation with you regarding animal biotechnology. So, it's first of all, I congratulate the entire college management and faculty members for organizing this very relevant and timely webinar on animal biotechnology. So, I'm just trying to show my presentation here. So, can you see my slide? Is it visible to you? Recent trends in animal biotechnology. Hello? हाँ, kindly please respond. Can you see my slides here?
Yes, sir. It is.
So it's visible to you? Okay. बोलो ना क्या
Sir, it is visible.
आ जाओ ना तुम इधर क्या प्रॉब्लम है Yeah. So I think I'm now well connected. So once again, I want to confirm that my slide is visible to all of you. Please respond.
Yes, sir. Yes,
Yes,
Okay,
sir.
okay, now. Okay. Now I'm moving ahead. So my presentation is on recent trends in animal biotechnology. Unfortunately, I'd like to share with you that just after the implementation of NEP curriculum in my university and in my department, we have introduced this course in the UG 7th semester as animal biotechnology. Earlier, we used to teach just biotechnology in general, but in my university, we have a separate department, a center of biotechnology. So they teach general biotechnology. Botany department teaches this and plant biotechnology, and we zoologists have started animal biotechnology in our department. So I have been mandated to develop this course and teach in the department along with some other colleagues as well. So let's start with some basic introductory aspect. Most of us know that biotechnology is nowadays has become so much attractive and interesting field of knowledge that it attracts many and most of the students from life science background, especially at PG level. So many students aspire to join masters, MSc or MTech in biotechnology. Although there are many hurdles as well in India, especially where job opportunities are very stressed and strained. So let's see. Biotechnology actually it fulfills several requirements of different areas like in food sector, clothing, shelter, health, and safety. While when we talk about its broader areas, so it generally helps in exploring different areas of science and technology in general. When we come to the basic aspect, definition, how can we define biotechnology? So it's so simple with its term, that is biotechnology. That means it's actually a combination of biology with technology. The study of science of living beings with the use of science in solving problems or the making of products which are beneficial to mankind. So this way, this new or recent branch of biology emerged as biotechnology. Now let's move on to animal biotechnology. So here this slide uh, depicts uh, pictorial presentation as well as some textual things are here. So you can see this animal biotechnology encompasses animal nutrition, animal vaccines, animal breeding, embryo transfer, transgenic animal, xenotransplantation, and many, many areas. So I think no area, no field in life sciences uh, remains untouched with biotechnology. And when we talk about zoology or animal sciences, so biotechnology is almost everywhere. It's interdisciplinary science. It combines almost all branches, especially modern branches of life sciences and technology. And I would like to add here one more point, that after uh, the rise and application of art, artificial intelligence, AI, the application and prospect of biotechnology or animal biotechnology has again you know increased manifold. So here you can see in this slide, it combines biology with technology, and it has its importance in agriculture, medicine, food production, animal health, genetic enhancing genetic traits of many animals and animal products, supports biodiversity conservation as well, and it addresses many ethical concerns as well. So this is one example where we can see how animal biotechnology or researches and innovations in animal biotechnology can solve many important issues of human welfare. So this is one example. You can see uh, two fishes have been shown here. Two fishes have been shown here. Both are 14-month old. So left one is genetically engineered or biotechnologically engineered salmon fish, while on the right side you can see normal standard salmon fish. So biotech engineering has been used to improve animal size or animal yield or animal product. So here in this slide, you can see how biotechnology or animal biotechnology evolved during times, so historical evolution. In fact, one way or other, it started historically at very earliest. It started with early domestication, where human being used to improve the breeds of animals at different levels. Then they used traditional breeding to improve the yield and improve the quality of animals. Then a lot of genetic discoveries or advent of genetics also helped in improving the quality of animals, which were used for several purposes. Then again, time to time, new innovations, new advancements in science and technology helped in improving the quality of animals, the use of recombinant DNA technology, then cloning and transgenic animals, then many after application of many new technologies, it became necessary to have some guidelines to regulate many experiments, many developments. So regulatory milestones were also applied and came into existence for safety and ethics. Then, of course, when it became a regular practice, then many industries and companies came into existence for growth of biotechnology companies started in different parts of the world. And nowadays we have many biotechnological applications, including gene editing, cloning, and diagnostics, which we will discuss later on. So slowly and steadily, it's moving forward, and we are seeing many new technologies day by day. So this is again, this slide is again showing how from very ancient times, using different means and tools, we human being, tried our best to improve the animal qualities and animal products.
Yes, sorry, sir. Slide is not showing properly.
Not
Let's
sure.
show thing.
Uh, Slide is so that may be some net issues, but here, what kind of issues there? That slide is not fully depicted on your screen, or what's the problem? Can you share? Let me see in my system.
Okay, oh, it is visible. Okay, sir. Now it is up.
So when I make it full screen show, I think then there may be some problem. This one.
Now it is showing. Okay.
So can you see full slide, or any part is missing? No, just give me feedback.
Now it is showing.
It's clear now. Okay. So this slide is again, this is showing some latest advances in the field of animal biotechnology, like knockout livestock using ZFN, TALEN, CRISPR-Cas9. So CRISPR-Cas9, I will discuss it in detail in coming slides. So these things have become recent developments in biotechnology, especially this CRISPR-Cas9 genome editing has revolutionized biotechnology nowadays. So this one is again very interesting. So those who are studying biotechnology or animal biotechnology, so they should know, and it's really very interesting. So to study different areas of biotechnology, so some experts of biotechnology have given some color coding of different areas of biotechnology. So we have red biotechnology, where we talk about healthcare and medical application, then green biotechnology. So sometimes the color itself is self-explanatory, like green related to agriculture, yellow related to food and nutrition application. Dark biotechnology, of course, it's related with bioterrorism and bio warfare applications. Then light biotechnology, management of misused biotechnology. Blue, where we deal marine ecosystem. So similarly, you can see white biotechnology, purple biotechnology, brown, gold biotechnology when we use bioinformatics in biotechnology, gray biotechnology, uh, environmental applications, and pink when we talk about human welfare and leisure applications. So this is really very interesting. Now coming to key principles of animal biotechnology. So these are different principles which are very regularly, and these are indispensable areas in animal biotechnology. So I have especially shown this CRISPR-Cas9 gene editing technology, which has got lot of significance, and it has become, topic of the day or area of today's research in many advanced research labs. So, main principles start from genetic engineering, gene expression control, molecular markers, cloning concepts, embryo transfer, stem cell research, genomics and proteomics, then CRISPR gene editing, comparative genomics, and bioinformatics tools. So these principles are, indispensable in animal biotechnology, and nobody can do any research or higher studies in animal biotechnology without using all these basic principles. Similarly, when we talk about techniques which are basic or which are, you know very important in animal biotechnology, there are, there's no end of list, so the list is unlimited, but there are certain very modern, recent, and advanced techniques which are regularly used, especially when we talk about animal biotechnology. Always remember, I am focusing here on animal biotechnology. So you can see artificial insemination has been a practice to improve animal quality and animal yields. Then embryo transfer, IVF, not only in animals, but it's very regularly used for in humans as well. Somatic cell nuclear transfer, again, it is very important technique in animal biotechnology. Transgenesis has been very old one, but is still in use. Then marker assisted selection, where selecting animals for breeding based on genetic markers. So we select animals on the basis of certain genetic markers, and then we go for breeding. Now, come to DNA microarrays. Students, especially PG students, must be knowing this DNA microarray array technique, or gene array, or DNA chip, or many names have been given. This is again landmark discovery, and this technique has almost revolutionized genetics and biotechnology, where we can see expression profile of almost all genes at a time in one go. Similarly, polymerase chain reaction, PCR, or which is this was the discovery of Carimulis in 1993, and he got Nobel Prize also. So this technique, this technique is used in thermocycler. The instrument is called as thermocycler. So this has again revolutionized many genetic and biotechnological experiments. Then genome sequencing, we all know this genome sequencing has been routinely carried out earlier as well. But after starting human genome project, which was completed in 2003, this genome sequencing has got more attraction and importance in animal biotechnology. And then we talk about RNA interference, where we can silence any particular specific targeted gene or regulate specific genes. So this is example of DNA microarray, where this is so much impressive and effective technique, where in a very 1.25 square centimeter chip, we can assess the expression profile of entire genome of an organism, all the genes of an organism. So this is really, if anybody is interested, and I suggest that the students and teachers in life sciences must study this, what is DNA microarray technology, and how uh, this can help in the field of research, in the field of biotechnology, especially when we talk about expression profile of genes. So similarly, RNAi pathway, where we can target, we can silence any particular gene in a cell, in an organism. And here I have shown this PCR, how amplification of the particular segment of DNA can be very speedily carried out, and we can get amplified copies of any DNA sequence or any DNA fragment in millions and billions within few hours only, which were very difficult earlier before the discovery of this PCR, which we used to do through cloning in bacterial system and other systems. So now it has become so handy, so easy. So when we talk about the scope of animal biotechnology, everybody knows that it helps in food quality control, analysis of milk and other animal products, development of disease-free animals, and an application of scientific and engineering principles to the production and processing of materials. And finally, genetically engineered animals can be used to develop new medical treatments. So actually using application of animal biotechnology is not only the welfare and yield production and quality improvement of animals only. Rather, we can have such animals which help us, the biomedical scientists, in in diagnosing, in exploring many unexplored biological biomedical issues as well. So many genetically modified animals are used in biomedical researches. So actually, when I used to teach my students, I always tell them that generally sometimes it appears that we are doing some betterment of animals, or we are helping animals, or we are doing some good for animals. But in fact, we human beings are very selfish. Whatever we are doing, apparently it may look like helping animals, but actually finally we human beings are actually benefited, whether we are increasing the milk yield or meat yield, or any quality of animal, or we are developing genetically modified organisms or animals. Finally we are actually the beneficiaries, we human beings. So we are doing all these activities for our own benefit. So always remember, so sometimes it looks like we are doing a lot of things for animals. Even if we are doing some ethical, we are adopting, practicing ethical guidelines during research. But in fact, that finally ultimately helps the researchers and help the human beings. That actually these are beneficial for human beings. So just continuing the scope of biotechnology, it helps in livestock improvement like milk yield, increasing the milk yield and increasing the growth rate, disease resistance traits, improving fertility, better feed conversion, stress tolerance, production of specialty uh, proteins, special proteins, that is also called as biofarming, and modification of animal products like altering fat or nutrient content for health, and then conservation of endangered breeds and development of new breeds also. So there are many areas related to livestock improvement where biotechnology and biotechnological tools help us. So here this map can show you how much improvement in livestock or food production is required globally. So this different colors show you that uh, this is actually hunger map, and the dark red color actually mostly in Africa uh, continent. You can see many countries are there in Africa where more than 35% population uh, comes under very high hunger index. So, or they are undernourished. Similarly, simple red color like Sudan, Niger, and other many other countries, and that is uh, India and Mongolia and Bolivia in South American continent. You can see in these countries also moderately high, undernourished people are there. So then moderate, then comes moderately low, where you can see orange color and yellow, and less than 5% where any almost there is no hunger problem. So still globally, people need food, and of course for that we are bound to improve food production, and for that animal food products are very important, and for that we need the application of animal biotechnology to improve livestock improvement. And then when we talk about animal health, again biotechnological tools help in the improvement of animal health. But always remember, I am very frankly sharing this view, my view with you, that it again here it looks like we are we are loving animals, but finally, actually, we all such activities and efforts are to have healthy and smarter animals for our own benefits. So, for that purpose, for vaccine development and diagnostic tools, therapeutic proteins, monoclonal antibodies, disease modeling, control of zoonotic diseases. So, zoonotic diseases are, diseases which are transmitted to humans through animals. Then, early detection of pathogens, personalized medicines. So, although this concept is for humans, but that is suitably fitting animals as well. Antimicrobial peptides and improved veterinary drugs. So, a lot of areas are there where we use animal biotechnology to cater animal health perspectives. Then comes the number of transgenic animals. So, use of genetic engineering and biotechnologies helping us to develop transgenic animals. So, again, here, transgenic animals are actually finally helping us, helping human being.
Sir,
So,
sorry for interrupt. Slide is not moving.
again, slight problem is there.
Yes,
Okay.
sir. Yes, yes.
Hmm. Is it visible now?
Yes, sir. It is visible now.
Uh, Now tell me.
Yes, sir. It is visible.
Okay. So can you are you seeing full screen, or is it full screen?
No, sir. It is not a slideshow, but
Not full screen. Okay. क्या सर नेचुरल बोला तो मां समझा So I am just making it full screen. Mhm. Okay, can you see now full screen?
No, sir.
Not yet.
Yes, sir.
It's not full screen.
No, sir.
Could you see now? How is it visible to you? Please share. Full
Yes, sir, it's visible, but not full screen.
refresh करो. How is it?
No, sir, not yet.
So can you people read in that mode, which is visible?
Transgenic animal, sir.
Yeah, yeah. So is it readable to the audience sitting there?
Yes, sir. Readable, but
Yes, sir.
not moving slides.
Now, just to check, has it moved now? ये क्या हो गया?
No, sir. No.
तो move ही नहीं कर रहा. अब यहाँ जाकर मूव करें. Has it moved now?
Yes, sir. Cloning in animal
Cloning
biotechnology.
animal. Now,
Ye-
has the slide moved now?
No, sir. Cloning in animal biotechnology is showing.
It's not moving further.
Yes.
इस मोड में नहीं हो रहा, तो चलो उसी में कर देते हैं. Yeah, I think now the problem has been removed. So can you see now slides moving?
Yes, sir. Yes. Yes, sir.
Okay. Sorry for disturbance. Sorry for technical glitch. So now let's discuss about cloning in animal biotechnology. So cloning has always been a fascinating area in genetics, in molecular biology, in genetic engineering and animal biotechnology. So and especially after the development of Dolly as clone, it has got more attraction and significance. So cloning is producing genetically identical animals from a donor, and for that, some basic technologies or techniques are there, like cloning techniques, and then the best example is Dolly the sheep, the first mammal cloned from adult cells, proving the techniques feasibility. And in agriculture, cloning elite livestock for constant traits. It is used for species conservation, elite animal replication, identical models, health concerns, and but its success rate has always been a big hurdle. Not all pregnancies result in healthy clones, and even when you see the story of Dolly, uh, when we talk about Dolly, so we know that Dolly died before, in fact, before time, and it developed many, health issues as well. So a lot of other advanced biological concerns and ethical concerns are always related with cloning. Now come to biotechnology in aquaculture. So in aquaculture also, especially when we talk about fish and some other marine, especially marine food resources. So in that area, again, biotechnology has almost revolutionized aquaculture products, improving, starting from improved fish breeds, then disease-resistant seafoods, and for faster growth, better feed utilization, which is required for other animals as well, and genetic monitoring for ensuring healthy and diverse populations, and developing transgenic fish. One example I can quote here, not only for aquaculture, but developing transgenic fish, like when we talk about biomedical research in using animal models. So zebra fish has become a very good model, and nowadays, and we all know, and those who do not know must be knowing, that the early developmental stages up to 3-4 days in zebrafish embryo remains transparent. Its entire body is transparent, and the internal organs and movement of drugs, movement of bacteria, and all internal structures and internal activities can be monitored through naked eyes. So, what people are trying in that case, we are trying to develop such transgenic fish, transgenic zebra fish, which may remain transparent up to its adult stage and for whole entire life. So that type of development will lead to breakthrough discoveries in many biomedical researches. So such things are going on such researches are on the anvil, and in coming days, in near future, we hope that we would be able to develop such transgenic animals which can help us solving many complex biomedical problems. Similarly, biotechnology can help in the development of sustainable aquaculture, improving water quality, and traceability means biotech systems track products from farm to table. Actually, now translational research has been given more importance. So whatever we are doing in the lab should reach to the field, and that product should reach to the table, means to for our to our home, on our dining table, in our kitchens. So this traceability means here, farm to table means what developments, what products are there in the field, in the farm, that and how those products are reaching to the table, we can trace. We can use DNA fingerprinting, we can use DNA barcoding, and we can use many such technologies which can trace the entire movement of such products. So again, regulatory case studies and regulatory issues are always there, and that has to be taken into account. Now come to the poultry biotechnology, application of biotechnology in poultry. So now I think I have taken much time and I would like to move faster. Many things are very clear to you. So we can enhance egg production, we can develop disease, and these things are in practice nowadays. So this is not just imaginary things. We have achieved many things in the area of poultry. That's why you can see a lot of chicken products are there in the market. So disease resistance, faster growth lines, genetic conservation, sex determination, improved meat quality, vaccine development, nutrition optimization, embryo technology, monitoring, and again traceability. So many biotechnological tools have already been applied and we are getting the results and outputs as well. Now come to the genomics. So as discussed in the beginning, that although genomic activities, genomics research, researchers were carried out earlier also, but after a human genome project, this has got more, significance, more importance, more attraction. And before human genome sequencing, we had already sequenced many other organisms, and even during the Human Genome Project, we developed the sequences, entire genomic studies of many organisms. And after that, again, so we have reached up to that level where it may be surprising to many, newcomers in the life sciences that now we are talking about human microbiome project that has been completed and its application has been started, where we talk about not only human genome. We talk about an individual, a human being, harboring or having uh, you know many millions and billions of microbes together. So we are not one entity, we are not one individual, one organism. All human beings are a set of millions and billions of microorganisms together. So millions and billions of microorganisms are living on our body and inside our body, and we are the result of the combination of all these organisms. So all these organisms are either affecting us in positive or negative senses. So animal genomics again has led to the development and improvement of many animal aspects, animal related aspects. So genome mapping, functional genomics, comparative genomics, SNP identification, is single nucleotide polymorphism has again led to the diagnosis and problem solving of many genetic diseases. And genetic linkage mapping, marker assisted breeding, genome selection, expression profiling, where we use DNA microarrays, and data analysis tools where bioinformatics and AI has again helped. So AI, we all know that in AI we use data analysis, machine learning, and finally artificial intelligence. So practical breeding benefits, where we use targeted animal improvement, and these many things have already developed and they are in practice today. So another important aspect is a stem cell in animal biotechnology. So stem cell biology, stem cell discoveries has again helped and revolutionized animal biotechnology, where we can use embryonic or pluripotent or adult stem cells. This is used. So always remember, animal biotechnology is not only helping animals. So many animal biotechnological developments are ultimately helping human lives and other areas as well. So it's used in genetic regenerative medicines, disease modeling, gene targeting, tissue engineering, cloning via stem cells has become routine practice, and production of transgenic animals, which I just talked earlier, and organ regeneration. So and then again, in most of the genetics, animal biotechnology, or any modern life sciences, biomedical areas, so always ethical and legal, LC, it is called as LC, ethical, legal, uh, uh, ethical, legal, and social implications. So LC is always there in all modern biological researches. So that has to be taken into account. Then coming to conservation biotechnology, it has become you know latest modern area of in life sciences and biomedical sciences, where we talk about this is in applied in wildlife sciences as well, where endangered species conservation requires many biotechnological processes. It's used we use cryopreservation, then assisted reproduction, reintroducing extinct genes, genetic rescue, gene banks, cloning endangered animals, monitoring diversity. International collaborations are required for such conservation. And even we need biotechnological applications for habitat restoration. So you see, we have genetic diversity in different organisms in the population. So many areas require conservation because rampant exploitation of many organisms for our own benefit has led to depletion and extinction of many living organisms. So conservation is again required. So when we talk about animal nutrition, again, biotechnology is helping us a lot in improving nutrition, help with the help of nutrigenomics, functional feeds, enhancing digestibility, lower environmental footprints, and probiotics and prebiotics for better gut health. So this is helpful for human being as well. And nutrient absorption, genetically modified feed efficiency, and personalized diets, metabolism monitoring, safe feed innovation. So many areas of animal nutrition requires biotechnological applications and biotechnological developments. When we talk about drugs, pharmaceutical production in animals, so of course this is again not on for only for the benefit of animals. We have developed bioreactors and biofarming. This is again very important, where transgenic animal uh, produce therapeutic agents like insulin hormones. So these we can develop such animals which produce a large amount of insulin and hormones, which finally we get for our own benefit. Similarly, antibody production, hormone manufacturing, vaccines, protein synthesis, milk as drug vehicle, we can develop such animals which produce milk having many pharmaceutical substances in that, so that milk will become a drug-like thing. Then regulatory challenges are again related to that. So LC will come into a come for play here. Market overview. So commercialization of pharmaceutical animals are also important. And finally, again, we come to the main point, that is for human health benefits, so treatments and diseases and improved therapies for human beings. So when we produce such animals which can help in pharmaceutical development, finally that will be beneficial for us also. So now coming to ethical issues and concerns, which I discussed several times. So all modern researches and advanced researches require fall force us to follow special guidelines developed for us. So in most of the biotechnological researches and studies, we have to know ethical issues. So there is CPCACA, and a guideline developed by Ministry of Environment and Forest. So we have to follow that guidelines for any such research in animal biotechnology. When we talk about genetically modified organisms, or we talk about cloning, or we talk about genetic engineering. So many social and ethical issues are there, and that has to be taken into consideration when we do research in uh, using animals and application of animal biotechnology. So this is very important. Earlier it used to be three R's. So now we have 4 R's in animal. Whenever we do researches on animals, so this is actually ethical guideline. We have to we should try to reduce the number of animals sacrificed for animal research. Then we should try to refine the research in such a way that animals are number of animals killed or sacrificed in research is reduced. And then replacement, we should try to replace the live animals, especially vertebrate animals, with some other animal models, research models, so that number of animals killed may be reduced. And finally, a new, this is the fourth one, is the addition in 3 Rs, that is rehabilitation. Many animals which we use during clinical trials, or we use in research in life sciences or biomedical sciences. So after doing research, we should take care of that animal. We should try to rehabilitate that animal for normal life. Now come to, we are almost coming to the end of this talk. So there are certain very recent advances and innovations in which I talked in the beginning. CRISPR-Cas9 genome editing. We are able to edit the genome, even our own genome. So designer baby concept has come. In many countries, people are trying to edit human genome in such a way that we can have desired genes in an animal or in human being, and we can have the baby or offspring of animals with our designed genes, with our genes, whatever we desire, whatever we want. So such cases are called as, designer baby development. So this CRISPR-Cas9 technology has, enabled us to develop such babies or such animals, and the researches are going on globally. Now come to synthetic biology. Here I would like to add one point here, that now we are people have started in June last year. A global research project has been launched in UK, where people are trying to synthesize entire human DNA. I am repeating. Now we are trying to synthesize entire human DNA, so that we can further, people are scientists, uh, generally we hear that, we all know that what is a fiction today can be reality tomorrow. So people are trying to synthesize entire human genome, entire human DNA. And that project, with the, collaboration with Wellcome Trust, that project has already started in England, in London, just in the June July month of 2025. Similarly, among the recent advances, we have xenotransplantation. So we are developing such humanoid organs in animals which can be transplanted into humans. Similarly, genome-wide studies are again leading to solving many genetic diseases. Next-gen sequencing has become routine practice nowadays. Nanotechnology is again helping us in many latest innovations and discoveries in animal biotechnology. Epigenetics has again become very important, where uh, the genome part, which were earlier considered as, junk part or junk gene, now these all have become very important and significant. And this uh, epigenomics has become a new branch of genetics nowadays. So similarly, personalized animal medicine, where we talk about, particular animal, individual animal, and tailored intervention or tailored medicine or tailored pharmaceutical products may be produced for a particular animal. So that will be called as personalized animal medicine. Then smart sensors have been developed with the use of, chip technology and nanotechnology, devices monitoring animal health and environment, and automation using robots for rearing, breeding, and monitoring. So in many very latest, and you can add AI. So in all these technologies, AI is helping us a lot. And AI will become, uh, uh, a very important tool in coming days, where it will be indispensable for all biotechnological research. Now, coming to challenges and limitations, despite all developments, advancements, and modern researches, we have a lot of technological or technical hurdles. And that will be, of course, that will always be there, and we will try to eliminate or remove or solve those hurdles, then still today technologies are there, but many and most of the technologies are having very high cost. So that has to be again addressed. And low public awareness, many new technologies, new developments are not known to general people. So awareness is also required. Then ethical legal complexity is always there, and unintended effects may or may not be there when we play with the genes, and when we develop genetically modified organisms. So there may be some off target gene changes, and unwanted effects may be there. Then regulatory variability in many countries of the world, different types of regulations are there, guidelines are there, so that has to be uniformly addressed. Then many advocacy group resistance, pushback from animal rights group. Many animal rights groups are active, and they create many issues related to animal biotechnological research. And many poor countries or developing countries have very limited access to such resources and infrastructure, so that issue has also to be addressed. And lot of environmental risks are always there to our ecosystem, and actually we need interdisciplinary collaborations across different fields to address many biotechnological issues and research. Here you can see what are the future prospects. So animal biotechnology market is growing like anything. So in between 25 to 30, it is expected to grow 9.9% global market. And you can see this animal biotechnology market from 20 to 2030. This is expected a rise in the field of market. So this slide is also showing you many global companies which are involved in biotechnological products. So many new emerging biotechnological tools, fields are there. Integrated management. This is again a feature prospect and feature challenges. Advanced transgenic lines, precision breeding, climate adaptation, food security, disease eradication, sector collaboration, and societal acceptance. So still many people are afraid of many genetically modified organisms or products. So that has to be solved. And one health concept has developed where we talk about animal human environment. So this is again latest concept, one health concept. So a student must remember that when we talk about animal, human, and environmental health together, these three are dealt together, then this is called as one health. So this is for students. If any student is uh, studying animal biotechnology, he or she may have prospects in many areas like pet and related accessories, meat industry, and then companion animal business, dairy industry, or milk processing, disease investigation and control, wild animal resource management, fishery production and trade, in pork trade also, livestock engineering and biofarming, animal gut microbiome. So any production industry, many biotechnological research areas, genetic research areas, animal healthcare, poultry sector. So many nowadays uh, globally people are focusing on animal products, and in that area animal biotechnology is helping. So those who have background or knowledge or research in animal biotechnology, they have a lot of career opportunities waiting for them. So these are the references, and with this, uh, thank you very much for your patient hearing, and I think so.
Thank you, sir.
I think this
Is
is
there
all
any
about my
question?
side, and if anybody has any query, any question, or any clarification, I would like my best. I will try my best to resolve those issues. I think the things are clear to all of you.
Is there any question? You can ask. Yes, sir. Yes, sir.
You just ask if anybody is asking, trying to ask any question.
Hello.
I have a question, sir.
Yeah. Ask your question.
How artificial intelligence acts as a game changer in animal biotechnology today?
Just pardon. Just repeat your question.
How artificial intelligence acted as a game changer in biotechnology
Artificial
today?
intelligence. Okay. So AI can help us in many ways. So start from just a study. In earlier times, before AI, we used to, uh, for getting any information, we were forced to give.
Sir, voice is not clear.
My voice is not clear.
Yes, sir.
So this is again, technical problem. So how can I help you? So whatever you can hear, you can try to understand. So in earlier days, we used to depend on books and research papers and publications and on the experts only. But with AI and many modern resources, we can have access to global information within fraction of seconds. This is one aspect. Another help which we get from AI is that uh, the data in life sciences, especially in biotechnology, we have huge data about genomes, about different genes, data about diseases, and data about many researchers. So analyzing these data manually is almost impossible. So earlier also we used to analyze different data, especially in genetics and genomics, using computer, just bioinformatic tools and computer. But AI has helped this bioinformatics again, and now the data can be analyzed, can be interpreted, can AI can give us desired result within fraction of seconds, within very less time. So AI is helping in all the areas of modern sciences, modern technology. But in biotechnology, again, we get the help of AI very much, and we cannot deny, we cannot, we have become dependent on AI nowadays, in especially biotechnological research. Is it clear to you? Could you get my point?
Yes, sir. Thank you, sir, for such an informative. Thank you, sir, for such an informative and insightful lecture. It truly enriched our understanding of the subject. Thank you so much, sir. Now, due to some issue, I conclude today's national webinar. Now, I will request Professor Javeria to propose the vote of thanks. Respected Principal Ma'am, Professor Dr. Mrunal Bhardwaj Ma'am, Vice Principal Dr. Arif Anjum Sir, and our esteemed resource person, Professor Dr. Hamad Ahmad Shadab.
respected members of the IQAC, faculty members, coordinators, and organizers, and all participants. Good afternoon to all of you. I am Professor Jairia from Zoology Department. On the behalf of MGV's Arts, Commerce and Science College, Malegaon City Autonomous, I take this privilege to propose the vote of thanks for today's one-day national level webinar on innovations and applications in animal biotechnology. First and foremost, I express my deep sense of gratitude to our respected principal, Professor Dr. Mrunal Bhardwaj, for her constant encouragement, guidance, and inspirational words, which always motivate us to organize such meaningful academic events. My heartfelt thanks to our eminent keynote speaker, Professor Dr. Hamad Ahmad Shadab, for sharing their vast knowledge, research insights, and enriching experiences. Her experts' lectures truly enhance our understanding of recent innovations and applications in animal biotechnology and made today's session highly informative and inspiring. A special word of thanks to all the participants who joined us from the difficult colleges. Your active participation, keen interest, and thoughtful questions added great value to this webinar. With these words, I once again express my sincere gratitude to all of you. Thank you very much. Over to Professor Sayyeda ma'am.
Thank you, ma'am. Thank you all for your valuable presence and cooperation. Have a wonderful day ahead. Thank you. Thank you so much.
Thank you very much, and congratulations to all of you.